All right, bio children, welcome back to another Screencast by Ms. Barlev. I know you guys must be so pumped. Okay, so you guys need to have, if you haven't already, you should have already watched meiosis first, okay? So if you haven't done that, you're in the wrong order. Don't confuse yourself. Go in order. Today is the genetics journal assignment, okay? So with the genetics, oh gosh, that color does not look good. I apologize. If you haven't noticed, nothing's changed. I still kind of talk to myself while you guys take notes. So here we are. If you guys took a look on Neo, you guys should notice that with the genetics journal, you actually are going to be taking these notes and turning them into Google Classroom. So as you can see here, for this topics worksheet, you'll be taking notes on these slides. You can do this in Google Classroom on the Google Doc. I've given you one in the, I don't want that anymore. Perfect. I have given you one in Google Classroom, so all you have to do is click on the assignment and click, and you can start typing. I know that's kind of hard with the screencast. What I've also given you is the old way I've done this, which is by actually making a booklet. Up to you. You can also just listen to this and then take notes. I'm writing all these things down for you guys as options, so it's completely up to you. So another option would just be to listen to this and then take notes. Completely up to you. The piece of paper way I just liked because you could always have it out and reference it. And that's part of what I would allow for you guys to use on the test. And so if this way works for you, feel free to keep that going. If not, just listen to this, sit back, relax, enjoy the ride, and then afterwards take notes in Google Classroom. But again, we should be doing this in Google Classroom. This is the assignment for this topic, okay? You'll also notice that when I have you guys do the Google Doc, that you guys are going to be doing a table of contents and writing things on pages. So this does actually mean individual Google Doc pages. Why do I have you guys do it this way? Just to separate it out and make it easier for you guys to go back and reference. This also allows you guys to insert pictures and have space to do so. So make sure you guys are doing that as well. Pictures are really helpful personally for me. So if I were you, I would be screenshotting pictures or something like that. So you guys can insert them into your own notes as well. A lot of this section here we're going to be doing is basically just vocabulary you guys need to know in order to understand what we're talking about. It's not actual content. It's not like any of the problems you're going to see. Obviously, I'll be testing you on vocabulary, but this is just basically the precursor to being able to do anything. So let's go ahead and just get started. So genetics, what are we talking about this chapter? This is biology that studies how traits are being passed on from parent to offspring. This is what I studied in college quite a bit. I actually took every single genetics series that was offered at UCSB. They have different perspectives. So genetics is very versatile. You can look at it from the molecular level. You can look at it from the population level, evolutionarily speaking. Lots of different options. So I really love it. If this topic speaks to you, go ahead and join Zoom and talk with me. I would love to geek out with all of you. The next term we're going to talk about is very similar to genetics. It's heredity, okay? Difference here is that it is just the actual passing of traits from parent to offspring, okay? Literally just this intermixing of mom plus dad gives you genes. That's heredity, and that's it. Genetics is the study that also involves the molecular level, how the DNA interacts, how you actually get the DNA into the cells, like meiosis, stuff like that. It's the whole picture. Heredity is just parent to offspring trait, okay? DNA, this should be a review. Deoxyribonucleic acid. And this is the molecule that contains the genetic information for any organism. It's always in the nucleus, can never leave. Protein, these are the molecules that are responsible for telling cells what to do. And again, these are how traits get expressed. So if you'll recall from last chapter, right, with the central dogma, okay, so reviewing central dogma, we have DNA that gets converted into RNA, which makes a protein, which expresses a trait. Notice how all this stuff is starting to connect, okay? So if you'll notice here now, okay, it says vocabulary page 2. So if you guys are following along and doing this, this is page 2 of that Google Doc or page 2 of your notes, okay? So next term we're going to learn is a gene. You probably have heard this a lot because everyone talks about different genes and genes that you have or don't have, stuff like that. A gene is a singular piece of DNA that codes for a certain trait. Now notice, like in this picture, okay, this whole segment, okay, or rather, let's actually undo that. This piece of the DNA is one gene, okay, one gene. And that might code for the example, like a trait that says that you can actually like roll your tongue. If you guys don't know what that is, if you can make a taco tongue, that's actually a trait that's passed on. Not everyone can do it, so you can go ahead and sit at home and try rolling your tongue. I can roll my tongue, but say for example, that's that segment of DNA that controls that. That is the whole length of it. It only does that one gene, no more, no less. Okay. Now this term is probably pretty new to you. You may not actually have heard of it, and that's totally okay. It's an allele. Okay. So al and then allele is how you pronounce it. Allele. Okay. It's different versions of a gene. Okay, so for example, it could be having scales or no scales. For the example I just gave you guys, it would be a rolling tongue, or you can't roll your tongue. Either one. Okay, so the alleles are the version. The gene is that segment of DNA that's going to code for whatever trait it may be. Staying on page 2. Sexual reproduction shouldn't be anything too new here. It's the process where two organisms contribute DNA to make a new organism. Genetic inheritance occurs here. Each parent has 2 copies and they pass on one to the other. And again, this is genetic variation. One of the 3 ways. Perfect. So say for example, okay, this is just an example slide. We have mom and dad. Dad has some characteristic, disease, trait, whatever it is, but he has it. Okay, that's what affected means. Mom doesn't. Okay. So they have kids, and they have 2 affected kids and 2 non-affected kids. Notice how it's literally just the combinations of chromosomes. So if we take this chromosome with this one, we get this affected kid. This chromosome with this chromosome, I'll do it in a different color. You also get an affected kid. But if dad passes on this chromosome with any of mom's, you get 2 non-affected kids. This is essentially what we're doing in this chapter. We're just going to be modeling it differently and expressing it differently. This is a very visual, hypothetical way, okay? New page. Make sure you guys don't forget. Okay, 2 new terms. These have been tossed around in the last chapter. This is going to make a lot more sense now in this one, all right? We have genotype and phenotype. If you'll recall, I said genotype is like gene. This is the combination of alleles an organism actually has. So this is what your DNA looks like, okay? What 2 versions of that gene you have in the 2 chromosomes that you have. We represent alleles with letters, okay? So if you'll recall from up here, I said that you rate them with letters. So here, I said if we had scales, it'd be big A and little a would be no scales. This is just for the sake of abbreviation. Does your DNA actually have the letters A and A in it? No. Do your DNA actually have any letters in it? No, it's all molecules, right? But this is how we express it in the science world, okay? So when you write a genotype, it's always going to be a combination of 2 letters. It could be 2 big letters, a big letter and a little letter, and a little letter and a little letter. Those are all the different combinations, okay? And what that tells us as scientists is which versions the parent has or the offspring has or whoever we're looking at has. When we're talking about a phenotype, okay, I want you to think like physical trait. I'll go ahead and add that up there for genotype too. Oh, shoot, I wanted to write gene. There we go. So when you want to express something's phenotype, what you're going to actually say is what it looks like. So what I'm going to do here is I'm going to say if it has scales or no scales. I can also say like blue eyes. I can say brown hair. I can say it has A blood. All of these are physical traits. And so whatever physical trait it is, that's how you express that phenotype. So these go hand in hand. You can basically say the same thing for them. But if I'm asking you for a phenotype, you're going to tell me what the actual organism looks like. If I'm asking you for its genotype, you're giving me the 2 letters its DNA has, the 2 different alleles. Okay. Now here, we have 2 terms that you probably have heard, understand, but in terms of genetics, there is a huge misconception, and so I want you guys to really pay attention to this. A dominant version of a trait is the form of a gene that, if present, will always be expressed because it has the power to mask the presence of the other, okay? Now, what that means is basically when you have that version, your DNA says, I'm going to express it, and it's going to turn off the presence of the other version. So we symbolize this with the capital letter. That version is big A or big B or big C. Does that letter matter? No, it does not. It is all arbitrary. I usually give you letters just to make it simple. But if I don't, you can make up your own letters. I just recommend, so like, think, for example, big A, little a, there's a very clear difference. If I did big S, little s, and I didn't have really great handwriting, right? Notice how it's really confusing which one's which. So pick letters that have clear differences. Okay. Recessive is a version of a gene that will only be expressed if 2 copies are present. So in order to express that version of the trait, if it's recessive, I have to have these letters. It could be little b, it could be little d, it could be little x. I don't really care, but that's how you express it. You have to have 2 copies of it in order for it to be present, in order for the organism to show it. Now, the misconception here. Dominant does not equal most common, okay? I'm going to say that one more time. The dominant version of a trait does not mean it's the most common. Why would people think that? Again, you only need one version of a dominant allele for it to be expressed, right? So if you have one, you're going to express it. If you pass on that one, you're going to have it, whatever. Probability says more than likely the dominant will have it. This is not the case. Dominant just means it has the power of masking it. If the dominant, right, is a bad thing, say it's like a disease and someone dies from it, right, the population's not going to have more dominant because they're all, I mean, not to sound morbid, dead. So don't associate common or popular or even like a benefit with dominant. I need you to just think dominant is what's expressed over the recessive. If you have questions on that, again, please join a Zoom session. I'll clarify. Now, I know this is a huge vocabulary blast, but it's so helpful to have this written down, typed down in one place, and then just be able to reference it so you guys can keep coming back to it. Homozygous and heterozygous, okay? I want you to think of the root word homo as same. And I'm sure you guys know the word like homosexual, right? You are attracted to someone of the same sex. There's other words like it, but I'm sure that's just the one that you guys know the best. Same thing with hetero, meaning different. If you're heterosexual, you are attracted to someone of the opposite sex. Most common example, there's a ton of other words that use these roots, so I don't want you guys to think that that's like all I have in my brain. It's literally just the easiest example, okay? So when you're describing a genotype, again, all these terms are for genotypes. If it's homozygous, it's 2 of the same letters. So you have 2 little a's. This could be homozygous recessive. And you have 2 big a's, and that would be homozygous dominant. If you have a heterozygous, there's only one way to get it. It's one of each. That's always heterozygous. There's no other way to have a heterozygous genotype. Okay? And again, these are words that describe a genotype. If I told you verbally I have a homozygous dominant genotype, you could give me big A as the actual genotype. So make sure you guys can picture that in your brain. Okay, so now we're getting to actual content. So this dude, Gregor Mendel, the homie. Gregor Mendel was a monk, and he lived in a monastery, lived a very monk life. If you didn't know, when you're a monk, there's not really much you can do. You sit in a monastery, plain walls, you pray a lot, and you get 3 meals a day, and that's really about it. So what did he do? He observed some pea plants, okay? Like the snap peas you can buy at Trader Joe's. Now, when he observed these, he saw some interesting patterns, and that led to him to discovering genetics. Now, before him, a lot of people didn't know what genetics was. We didn't know how traits got passed on, stuff like that. What was assumed to be true is something called blending, where offspring is just a mix of their parents. So if I had blue eyes and my future husband that's non-existent had brown eyes, our kid's eyes would be a green. If my eyes, or I'm blonde, and my future non-existent husband has dark brown hair, our kid would have brownish blonde hair. If I'm 5'8" and my future non-existent husband is 6'10", a girl can dream, our kid would be 6'3". Okay, that's what they just assumed to be true. Obviously, there's anomalies way back in the day where parents and their kids didn't have that blending, and so they just assumed that to be mutation and odd chance. Maybe the kid's adopted, dad milkman type stuff. Because of Gregor Mendel and him studying peas, we actually learned how genetics works, and we were able to explain these phenomena. So let's talk about these peas. Why did he study them? Or how could he study them and actually make conclusive results? Well, first of all, he could actually select which plants to use. If you didn't know, farmers are able to cross-pollinate plants that they want to produce so that they have whatever traits that they want. So if you wanted a strawberry that's big but also really sweet, they would cross-pollinate the 2 plants that have the biggest and then the sweetest. Short generation time. Okay, if you didn't know, takes 9 months for a human to grow, and then about 20-something years, sometimes longer, for them to mature. With peas, we're talking weeks to months, so a lot easier to do. Lastly, he noticed that every trait had 2 versions without any intermediate. So again, it's not this 5'8", 6'10", 6'3" business or anything like that. Instead, we saw 2 versions. What do I mean by 2 versions? Let's take a look at this table here. Okay. So here are all of the traits that he studied. These are what he saw in the plants. He saw seed form, cotyledon, that's just another word for the seed, color and form of the flower in the pod, and then stem placement and size. Each of these have 2 options. So form, the seed was either round or wrinkled. For the actual seed, he says yellow or green. The flower was white or violet. The pod was full or constricted. Basically what that means is just the outside was kind of tight and you can see the seeds on the inside. Pod was green or yellow. The stem was axial or terminal, just meaning top or middle. And the size was either tall or short. So again, 2 versions of every trait. No middle, no other options. And so here's an example of what Gregor Mendel did. Say, for example, he wanted to look at the cotyledon or just the seed color. So here's the parent generation. He had parent 1 being yellow and parent 2 being green. What he noticed, and you'll see this F1, that just means first generation after the parents. You can abbreviate parents as P. What he noticed was all 4 were yellow. Interesting, one of the parents is green, so that's odd. You would imagine some of them being green. Let's see this again. They crossed these 2, and in F2, or the second generation, we had 3/4 yellow and 1/4 green. They repeated that step again, and we again notice this 3/4, 1/4 ratio. So if you had to take a guess, which color do you think is dominant? Yellow or green? We're going to base that off the definitions we went off of earlier, where dominant is able to mask the other, and recessive has to have 2 copies. So which one do we think is dominant here? If you said yellow, you would be right. Green is going to be the recessive. So here are Gregor's conclusions. Gregor concluded that inheritance of each trait is determined by units or factors that are passed on to descendants. These are your alleles. Gregor also discovered that an individual inherits one of each unit from each parent. This is sexual reproduction, 1 chromosome from each parent. The last thing he concluded was that a trait may or may not show up in an individual, but can still be passed on to the next generation. That's the difference between dominant and recessive. You have to have 2 copies in order for the recessive to be expressed, and you only need 1 for the dominant to be expressed. So based off of these findings, we now have 2 different laws that come from Gregor's experiences. We have the law of segregation, which is the first one, and this just states that the pair of alleles from each parent separate and only one gets passed on to each offspring. So if you look, here is this parent's genotype and here is the green parent's genotype. The gametes form and only one Y gets put into each gamete, and then they 2 randomly combine to form the offspring. So that's the law of segregation. For the law of independent assortment, these alleles being passed on into the new offspring is random chance. So there's an equal chance of all of them being added in together. All combinations are equal. Okay, so I'm going to have you guys pause here. You should do the colorful karyotypes assignment that's in Google Classroom now. When you're done, you're going to move on. So again, pause this video here. I want you to do the colorful karyotypes and then move on. The notes are going to keep going. Okay. But I want you to do the colorful karyotypes now because it just makes the most sense to do it here. Okay. Welcome back. Did you see what I did there? Welcome back. We're now going to move on into the simple dominance version of these notes, or basically the Punnett square is the real genetics deal here. Okay. So, simple dominance. This is an inheritance pattern for a trait that is coded for by a single gene. The trait has 2 phenotypes, a dominant and recessive, and the dominant hides the presence of the recessive. This is like the pea plants. So essentially what this process is how Gregor discovered genetics. It's the most simple form. We're going to move on to the more advanced forms later on in this chapter, but this is where we're going to start for now. So when you do a simple dominance problem, there are a series of 4 steps. You're first going to write down the parent's genotypes, which is the letters, okay? You're going to draw a Punnett square. Now, a Punnett square looks like this. This is how we describe, or actually more rather, solve problems involving genetics. The boxes represent each possible gamete a parent can contribute. So that would be like parent 1, and this would be like parent 2. I need to change the color here. And again, like the law of segregation, the law of independent assortment state, there's equal chances of them making it into the offspring, so we have equal sized boxes for them. You're going to write the parent's genotypes across the top and left sides. That's what I did up here. And then you're going to fill in the boxes by bringing the letters across and down. So I have an example problem here for you guys that we're going to walk through. For the purposes of just this first problem, I don't have any traits associated. I'm just going to show you guys how to fill in a Punnett square. So here's my Punnett square. And like I said above, I have two parents. I said big A, little a was 1. I'll show you here. And the second parent is big A, little a as well. Different parents, just same genotype, and that's okay, it happens. And it's really important that you fill them in on these sides. It has to be here and here. You're going to fill in across and down. Now, what we're doing is showing every possible combination that each allele, or gamete rather, could have with the other parent. So what I'm going to do is fill in down with parent 1, big A, little a, big A, little a. I'm going to do the same thing with parent 2, except I'm going to go across. Big A, little a, little a. Now, notice how each box has one copy from each parent. That's sexual reproduction, right? So that's a good check to make sure you're doing this right. Okay. Another key feature here is the capital letter always goes first. Okay. That's like with writing your name, right? My name is not Stephanie. My name is Stephanie. So the capital always goes first. So that's just how to fill in a Punnett square, okay? We're going to try a practice problem here. I'm going to walk you guys through it before you guys do this on your own, okay? So we have some dark stripes in zebrafish that are controlled by a single gene called MIFA. That's actually how it's controlled. Dark stripes is dominant to no stripes. When you do problems like this, I want you guys to highlight the dominants here, okay? So you guys know which ones you're working with, okay? We have a heterozygous dark striped fish mating with a fish with no stripes. And I want to know the possible genotypes and phenotypes of the offsprings and their ratios. So I'm going to highlight the question here as well. This is what I'm solving. So, where do I begin? Well, let's look at the notes. First, I need to write down the parent genotypes, okay? Well, I'm not given the parent's genotypes. I'm given their physical traits or phenotypes. But in the problem, I'm able to determine that based off of this. So let's look at parent 1. Parent 1 is a heterozygous dark striped fish. Dark stripes is dominant, and heterozygous, remember, is 2 different letters. So in order to note this parent, I'm going to say it's big N, little n. Heterozygous, hetero meaning different, dark stripes, big N, little n. Parent 2 is a fish with no stripes. Oh, shoot. There we go. No stripes. No stripes is recessive. How do I get a recessive phenotype? I have to have 2 copies of it. So I have little n, little n. Now I have my 2 parents' genotypes. What am I going to do? I'm going to draw a Punnett square. Here's my Punnett square. I'm going to write the genotypes across the top and left sides, keeping the parents together. So I have parent 1, which was big N, little n. And I have parent 2, which is little n, little n. Again, I'm going to fill this in across and down, so I have little n moving across, big N moving down, little n moving down. Okay, so my Punnett square filled in, but that doesn't answer the question. I want to know the possible genotypes and phenotypes and their ratios. So what I'm going to do is I'm going to write this over here. Let's use blue, okay? Genotypes. What are the possible genotypes of my offspring? It's any of the genotypes that are in my Punnett square. So one of them is big N, little n. One of them is little n, little n. Do I have big N in my Punnett square? No. So that's not a possible genotype. So I'm going to go ahead and even just like erase it. Okay? Those are my possible genotypes. What about phenotypes? That's not written in my Punnett square, but it was given to me in the in the problem. It's stripes and no stripes. You can't guess or make up phenotypes. I give them to you. So make sure you always look back at the problem. Now, when I'm saying ratios, I want to know how many of my boxes had each or expressed each. So big N, little n, how many boxes have it? 1, 2. 2 out of 4. How many boxes had little n, little n? 1, 2. 2 out of 4. 2 plus 2 is 4. 4 out of 4, 100%. They're all accounted for. It's another good check to have. Stripes and no stripes. Well, I need to know which one was dominant and which one was recessive. Dark stripes is dominant, no stripes is recessive. So, how many of my boxes are dominant? 2, and that would be stripes. So I'm going to say 2 out of 4. Little n, only way to get the recessive, 2 out of 4. That's what I mean by ratios. I don't want like 1 to 1. That's a big no-no. You guys are not going to do that right and just confuse yourself. Just count the boxes out of 4. Can you simplify this to 1/2? Sure. Do you have to? No. So maybe don't waste the time. Okay? All right. So now that you've made it through these notes, what you need to be doing is finishing the notes in Google Classroom, the genetics journal. You need to do the simple dominance review. And on Neo, you have some quizzes. You guys have, let me look for it. You have the simple dominance quiz and the genetics journal quiz. So, and that is it for right now. Once you finish all that, you're done with that section's work. Make sure you guys stay on top of things. Again, if you have questions, Zoom me, email me, Neo Messenger, all that good stuff. Nice, guys.